Hello everybody, I'm back. Dr. K.K. Agarwal, President, Confederation of Medical Association of Asia and Oceania, past National President, Indian Medical Association, and President, Heart Care Foundation of India. And you are watching my Talks with Dr. K.K. Agarwal, 7 p.m. show, 156th COVID edition non-stop. Let's have the headlines of the day. With less than 20,000 new cases, India records the lowest number of daily new coronavirus infections after almost 6 months. Taiwan today reported its first locally transmitted case of COVID-19 since April 12th. More than 100 contacts of the patients are now being tested. More than 40 countries, including India, have banned travel from UK because of concern about the new highly infectious COVID-19 501 variant detected in England with 23 possible mutations. Meanwhile, Pfizer and Moderna are testing their vaccines against the new UK variant. A door-to-door survey of 1.72 crore residents of Delhi has revealed that only 39.5 thousand of those surveyed had COVID-19 symptoms. Now, before I go to the subject of the day, it's a customary to learn a hand wash. Let's pick up a ultra protection gel lasting for 2 hours, and we take and we practice S for Seedha, U for Ulta, M for Muthi, A for Angutha, N for Nakhun, and K for Kalai. Once it is done, we are ready to go to the topic of the day. The topic of the day is 501V2, the new coronavirus variant in South Africa, and its difference from the one which is in UK. The UK one is more in the news than in the South Africa. UK one has 23 mutations, and the South African one has 3 mutations, but all 3 of them are in the receptor binding domain, which basically causes a concern to us. And out of that, one is in the receptor binding moiety. That means 3 of them in the receptor binding concerns us. The scientists will be able to tell which one is more contagious, but yes, the thing which is common is 501. And I have with me, let me introduce my guest today. She is Dr. Angelica Coetzee. She is the National Chairperson, South African Medical Association. She qualified as a family practitioner in 1985 and is now a full-time practitioner in private practice. She is a member of the Ministerial Advisory Committee on COVID-19 and long haul COVID. That means what we call them as post-COVID here. She is the recipient of the Clinical Excellence Award 2020 by Discovery Health. So all over to you, Dr. Angelica. Can you now present your findings and then we have a discussion?
Thank you. I will I share my presentation?
Can I have the slides, Ritu?
While we're waiting for the slides, I just would like to thank Dr. Agrawal for asking me to give a bit of a background of what we are currently seeing in South Africa. And I would also like to say a very hard welcome to all your viewers out there watching this show. I think you are very much in the forefront, Dr. Agrawal. And you are a leader by having this type of weekly or daily interviews that you are conducting, and I salute you for that. It's always an honor to be part of such a good.
Thank you.
Thank you. I would like to make first of all, before I'm starting, this is not my slides. These slides is coming from these specific ones from the work that has been done by Prof. De Oliveira of the CRISP Foundation in South Africa. And they started to look at the genomics as a potent tool in the COVID-19 fight. And the reason why they looked at the genomics is to discover what virus is causing the COVID-19. For instance, we all know it's SARS-CoV-2. But to look at your genomics, you also develop diagnostics like the PCR, and it is also important to develop your vaccines. Currently, we know there is 2 vaccines in the market. It's the mRNA and the vector vaccines. mRNA is the one that Pfizer is using, and the vector vaccines, for example, is the AstraZeneca one. To look at your genomics, it also helps you to track the transmission. And with transmission, we mean outbreaks, spread, and where it starts to happen. And also to identify if there's any reinfections, and then to understand the host virus interaction, which is extremely important in this case. So of all these genomes produced, only 2,500 is part of South Africa. Next slide, please. So just a very quick introduction I was showing you here. If you look at the first graphic of the epidemic and genomic data in South Africa, you can see that was from the first wave. This is important. We are now in the second wave, the first wave. You can see all the sequences there of the viruses. You can see our trajectory, how it went up slowly, the curve, and how it came down. And if you look at the introduction into South Africa, here you can see the origin of, this is from Africa infections, the blue ones. You can see Asia is the green one. A lot of them came from Asia, Europe, North America, and South America. So if you look at the sampling in South Africa, not a lot was outside of South Africa. Most of it was in South Africa. If you look at the next slide, so this is an important slide. If you again look at the first wave, here we had multiple lineages that was circulating, and it was 3 lineages here was responsible for 42% of the infection. And again, here you can see the lineages, you can see the green, the darker blue, and the lighter blue that we have seen in South Africa. Again, remember this is the first wave because our second wave is going to look differently. Can I get the second slide, please? So here is what we are currently experiencing. This is now the new lineage in our second wave. And you can see it's on the green, the yellow one, it's very dominant. And it accounts for more than 90% of the infections that we are currently seeing in South Africa. You can see the rest that is still there because people think or tends to think that once if you go into a second wave, the other variants of the virus is not there anymore, but they are still there. It's just that this new, I call it new kid on the block, is responsible for 90% of what we are currently seeing. Next slide. Here is what Dr. Agrawal spoke about, the importance of the glycoprotein and receptor binding domain. We have, I think it's common knowledge that the protein loves to attach on the ACE2 receptor on the host cells, and that causes a lot of inflammation. And then you have each of the 3 petals of the spike protein. It sort of carries a hook, and that hook is called the receptor binding domain, and that interact directly with the ACE receptor in the human cell, that little hook. And then again, the receptor binding domain is also the main target for neutralizing the antibodies and vaccines. And this is also very important. Currently, we don't understand everything or the extent to which this is happening. but it's important to understand that. And then again, mutations therefore might affect the ACE binding and or neutralization of that. And this is what we are seeing, the three bindings or the three mutations. Next slide. So here, the three mutations are at a key residues interacting with the human ACE receptors and with neutralizing the antibodies, as I have just mentioned. Here you can see the three types of mutations. It's the NO5 is currently our most important one, and it binds, it enhances the binding affinity, especially to the ACE2 receptors. Then you get the E484K, enhances binding affinity to the ACEs and also confers resistance to class 2 antibodies. And then you get the K417, and also likes to contribute toward immune evasion at this site. So I'm not going to go into a lot of detail. This is not my domain. This is the domain of Prof. de Oliveira, but I just would think that it is nice for people just to see the three binding sites and the domain and what we're talking about. Next slide. So from Prof. de Oliveira and his group at CRISP, the next steps that they've now identified this, and now the key task is to get a better understanding of whether there's any clinical and epidemiological evidence to suggest the phenotype impact. So at this stage, it's increased transmissibility, pathogenicity, and escape from the neutralizing antibodies so that reinfection can occur. They are working very hard in the background, again, to look at this. For now, what we are seeing is there is increased pathogenicity and transmissibility, and the infection of the virus in a cell system. They are also growing the virus and conducting experiments to assess the functional effect of these spike mutations. Again, as I've said, in ways of how the virus infects the cell system, and the neutralization assessed with antibodies from patients infected with other circulating strains without spike mutations. This is extremely important stuff, because this will help us to understand whether the vaccines that's out there will be efficient against this new lineage that we have currently and what we are seeing. A lot of this work is still going on. We hope that we would get some answers within the next 6 to 8 weeks. Next slide, please. And then what they are also busy doing in South Africa is they intend to intensify the sampling from other districts in South Africa. Remember, a lot of this sampling came from the Eastern Cape, where it was originally discovered, the new mutation or the new lineage was discovered there, and it rapidly spread up and down along the coast. I'll show you pictures of a graph of that. Unfortunately, due to the hard work and the difficulty of this, they haven't yet expanded sampling to other provinces, but they are now busy, and it's going into the Gauteng area, which is the most densely populated area, and it is what we always call the economic hub of South Africa. They would also like to link with clinical investigations of cases of suspected reinfections and severe disease development. Going on in the background. Again, it's a lot of work that is going on. Next slide, please. And the conclusion from Prof. de Oliveira and his team is that genomic surveillance is a critical component of the public health response. It's extremely important because it helps you to get early detection and then to track your new lineage, in this case, especially the 501. And what they also said is that this new lineage of mutations at key spikes in the glycoproteins, it seems that there's rapid and diffuse spread through the 3 provinces, and we have seen it. I'll show you some of the graphs that actually doesn't go slow, but quite a straight upward curve to the to the top. And the full significance of the mutations is yet to be determined, and work is ongoing to characterize the phenotypic impact. So it's still a lot of work to be done, but at least our scientists in the background started with this, and this is all groundbreaking work, especially in South Africa. So this was work that's been done by the CRISP Foundation with Prof. De Oliveira, and then we also have from the Caprice. You can give me the next slide. From Prof. Slim Karim. I've got some few slides. Next slide.
Next slide, Sarab. Ritu.
Thank you so much. Again, this is the slides that what is important here is you can see this was it's about a 7-day test, and if you look at this is our first wave. Very nice slow upward curve, and then slow downwards, and with our deaths that you can see there. And this sort of ended around about September, and we started here in early November, seeing a new slight upward curve and our trajectory that you can see, and it all of a sudden our cases started to jump. Now in South Africa, the next slide please. We look very closely at our cases. It's not that we have this huge amount of patients as you have seen in India, but 10,000 patients per day for us in South Africa is a lot of patients. Here you can see there was the first wave. You can see the curve was not the same. The Western Cape, this is recent slides up to the 17th of December. You can clearly see that in the Western Cape, the sort of straight line of the second wave. And this is most probably due to the 501 variant that we are seeing. And 90% is coming from that, from the 509 variant or the new lineage. Next slide, please. Here again, KwaZulu-Natal. You can again see the steep upwards movement of the second wave. Here was the first wave, and it follows a different trajectory than the first wave. Next slide, please. So there's a fine balance between virus and host. So very important to understand that your viruses usually evolve to become more transmissible and less severe. And it's mostly within humans in response to the immune pressure, and sometimes when they pass through another species like what we have seen, the bats and those type of species. And in the first wave, our SARS-CoV-2 was relatively stable. We recorded about 35 lineages. Most were the most transmissible D614 G variant from Europe with minor variations. But now in the second wave, in all provinces, there's early signs of spreading much faster than the first wave. It's not clear as yet, because we're still in the upwards curve, whether the second wave will have more or less deaths. I think that question would be answered within the next, 4 weeks, especially after the festive season that we have in South Africa. Next slide, please. I just want to show you again here the second wave. Here again, it's the 501 V2 variant. It started in Nelson Mandela Bay, in this town of the Eastern Cape, and it spread up towards KwaZulu-Natal and down towards the Western Cape. And again here, you can see this is the predominantly this new variant that is affecting 90% of our cases. Next slide, please. So again, we are it seems to us that the V2 variant is associated with a higher viral load, and that in the swabs that's been examined by the different groups looking after, looking after this and investigation that they have done, is you can see that the higher viral load in swabs that they now see may translate to the higher efficiency of transmission, and also might translate to a higher R0. And while other viruses are transmitting, this variant is transmitting faster. As I've said, there's still the other viruses are still there. Don't think they're not there. They're there. But this yellow one is the one that moves the quickest and seems to be the most infectious at this stage. And we will this slide was done about a week ago, where they were still speculated that this may translate into a second wave that will have many more cases than the first wave. I can say today that it is definitely, we're going to have more cases in the second wave than that we have seen in the first wave. Next slide, please. So we're nearly finished. So very important, this is also Prof. Slim Karim's presentation here, where he says, what do and don't we know about the variant? We know it's unusual for a new variant to contain several mutations, 3 in the domain. And the most important one is the N05, which alters the ACE2 affinity and most probably the antibody effects. It's also been reported in other countries like the UK, but important to understand, we are saying that we have the 501 variant 2. 501 in UK is not exactly the same than the 501.2 in South Africa. Again, early signs, that was when Prof still was preparing the slides, is spreading fast. We know that is spread. We do know that today that it is definitely spreading much faster than the first wave, and it is much more widespread. It's most probably across most of South Africa. Where did it come from and why did it form? We do not know. We do not know why it started in Nelson Mandela Bay. There's a few hypotheses that Prof. Salim was investigating. There's a scientific document that he's going to release within the next 2 or 3 days, where he gives a bit more background on some of his hypotheses around this. Is it more severe? Well, we are seeing, it seems to us that it is more severe. I'm now talking about from the medical practitioner side. And we do not know whether it's reinfecting people who got infected in the first wave. And we also don't know, as I've mentioned before, whether current vaccines works against this variant. And they are all being studied by the CRISP and the CAPRISA foundations. And what should we do? And I think this is something that what should we do next is something that's important to all other countries. It's not only related to South Africa. There's a reason for concern that we have a virus that seems to be spreading rapidly, but it is something we can deal with. And we keep on saying, Dr. Agrawal, I was so glad to see that you had this ritual of showing people how to wash their hands, because that's one of the ways. The mask is the other way. And the personal distancing between you and someone else. And then the ventilation, natural ventilation out in the open is the best place to be. And we are now trying to educate again our population that it's the same preventative measurements that we took in the first wave that is applicable into the second wave. You can see there's other variants in other countries, Australia, UK. We've all recently heard that the UK borders are being closed, or the other countries doesn't want to have people coming into their country from the UK. I think it's great, but the spread of this virus is so fast that I think it's already a bit late. But again, from South Africa's side, and I think it's also important to other countries, is that we had informed the key role players, and we informed the public last week on Friday early night, we started to inform our public, and we told them about this new variant that's out there. And then data, to publish data in a prominent journal of what we are seeing here. Then we need to urgently increase the phylogenetic screening and to sequence the viruses every month from across South Africa. And then we need to complete the studies to answer the four unknowns. Unfortunately, fortunately, there are vaccine trials underway in South Africa, for example, Johnson and Johnson. So what's going to happen? They are also going to ask them whether they can be part, and let's see whether we can, these vaccines can combat this new transmission of the new the V501 V2, which is extremely important. And as I've said, only the time will tell us. This is one of the problems that we have with these viruses. We try on a scientifically basis to think ahead and try to outmaneuver the virus, but this virus is, I think it is one ahead of us the whole time, and we need to play catch up. But at least we have the mantra, the four things that we need to do: the masks, the sanitizing, social distancing, and the ventilation. That's extremely important. And that is my presentation, Dr. Agrawal.
Thank you, Anjali. Can we have the slides off? It was a beautiful and elaborative presentation. I can expect only from people like you, who are with the government, and you are also in the advisory committee of the vaccine and a long haul COVID. And that's only we can expect from people like you. So coming back to, I want to compare South Africa with UK for the purpose of, because the fear people are having is everybody is after UK and ignoring South Africa at all.
Yes.
They are absolutely, nobody is talking about South African stage. Now it started on 16th of November. That means already almost a month. Do you think that all the second wave in all the European countries, because there are only two reasons for a second wave, either a behavior or a mutation. They can't be the third reason. So up till now, we were thinking it's a behavior, that because of the winter, the second wave is coming. Do you think that all these European countries where the second wave is cutting, actually it may be all a mutation? May not be, it may be only one thing which is common is 501. Okay, rest of the mutations are different. Maybe in every country they have some 501 which is mutating.
I think so. I fully agree with you. We are just lucky in South Africa to have very, very good scientists, and they started to work in the background and said, listen, this doesn't make sense. What they are seeing doesn't make sense. And they then started to do this genomic testing and looking at it, which is absolutely correct. And I think they were the first to actually alert the United Kingdom that there's a problem. And I would urge all the other countries also to go and look and see what is happening. I think the fastest spreading of the new mutations or the second waves that we are seeing, because if you look at most of the countries, the second wave seems to be much more virulent and most fast spreading than the first wave. And I think they will also find their own 501s, but different variations. And again, it's nice to close the borders, but I think to try to close the borders now to prevent the spreading of this 501 from South Africa and United Kingdom, I think it's already too late. Once if it's in your country, it's in your country. And you also need to go and look at your own mutations that you will have in your country.
Another thing which I feel is that in South African one, because we have two more mutations in RBD gene, and both of them are affecting the NABs, which is neutralizing antibodies. So maybe these types of mutations may make this virus, the person is going to get it again and again, and the antigenicity will be less. And these are the people who may start getting lots of reinfections. And that true antigenicity, NAB's portion is not there in UK.
That I'm not sure about whether it's the those two mutations with the antibodies, whether they are in the UK. I'm not that I don't.
No, I have all the
don't be
data of 23 mutations of UK, and these two are not there. In UK, all of them are in non-RBD. Only one is in RBD.
That is a huge problem then, because as you correctly said, we don't yet know what our antibody system is going to do. But it seems that these two mutations neutralizes the antibodies, and that might have an effect on your vaccines going forward. And as you correctly said, you can be reinfected. At this stage, as Prof. Salim Karim always says, is we don't know. We are speculating, and it seems the correct answers is what me and you are thinking about. But this virus is on its own. We can, you don't know. But it is actually very, very concerning that it cannot neutralize the antibodies, because it would have an impact on the rest of the world as well if we are picking this up in South Africa.
The clinical tip for high viral load is the guess we can do it with the cycle threshold value of the RT-PCR. Any study going on to find out whether the RT-PCR CT value is lower in the second wave compared to the first wave, or the incubation period has changed, or the infectivity. Normally, in a normal case, we have virus within 7 days, the virus get killed and the lymphocyte starts increasing. So are we finding any change in the wave 2?
So I know that, again, that Prof. De Oliveira, they are working in the background on this, and their findings will be published very soon. I have seen a preliminary draft of Prof. Salim Karim's document that he will also release, as I've said, hopefully within the next week. And it is extremely difficult to understand this virus going forward in the concept of will you see more cases? Will you see more severe infections? Will you have people, as you correctly said, being reinfected and reinfected and reinfected? Would it be the same than the flu virus? Every year you get a new variant. But something that is a bit of a concern that Prof. Salim Karim mentioned was that normally your virus tends to for the virus itself to survive, it tends not to become so virulent. But it seems that this 501 doesn't follow that pattern. It's still very, very virulent. And again, that is not in line with what this virus normally, or a virus behavior normally is.
So all the viewers, we have watched Dr. Angelique talking about the situation in South Africa. And she has come back with very basic points that South Africa and European countries are facing now a new mutation. Maybe that mutation is already there in other countries. We don't know. They will have to find out. One thing is very clear, and that is that this mutation is, the contagiousness is much faster, much bigger. And even if it is not more virulent, because of the mere numbers, and even if it is the same mortality, the number of deaths will be much higher, much more in this particular aspect. So we need to see more. So for the benefit of the people, we should concentrate, we should remember that there is a virus. The virus has a spike protein, and the spike protein has a component called RBD, receptor binding domain. Inside that RBD, there is a RBM, which is called as a receptor binding moiety. And there are 70 amino acids in that RBM, and they are from 318 to 510. And during, this is the 318 to 510 is the RBD, and 424 to 494 are the RBM. Fortunately, the 501 is not in the RBM, it is in the RBD portion. If this 501 could have been a amino acid between 424 and 494, the virulent form would have been there. We would have been in more trouble. And it is also, we are fortunate that it is, we are having a NY501 mutation, because there are other mutations which they have said is that it is a different, some other types of a amino acid change. It can change the further virulence of the virus. So let's go on watching. Let's trace, read, watch what is happening. We cannot tell about the mortality. Mortality comes after 2 weeks. We cannot tell about antibodies formation because that takes 28 days to form. So once the virus is more than 28 days old, in UK and in South Africa, we will be able to find out whether it is forming enough antibodies or not. But theoretically, there is a risk that this virus may not be forming that amount of antibodies as the first wave. There is a theoretical risk that this number of cases will be much, much, much higher than the first wave. Whether it is going to cause more mortality, we don't know. That's all for today. Let me thank Dr. Angelique from South Africa to be here. And as promised, we will bring you the experts from all over the world. We will try to bring another one from the UK to talk about the exact UK situation from the horse's mouth. So let me thank Dr. Angelique to be here with us. We are grateful to you to have presented this data, which would not have been possible without your help. Thank you, Angelique, and Namaskar. Namaste is one we call them as to, so that we don't have to shake hands. And we'll be back tomorrow, 7 p.m. with another show. Keep watching Med Talks with Dr. K.K. Agarwal. Stay tuned. See you tomorrow. Namaskar. Thank you, Angelique. Bye-bye.
Bye.